Hello class, good morning. Myself Vivek Yadav and I am here for class 12 biology, and the topic that we are discussing is biotechnology. And yes, sorry, because of some technical issue yesterday in between class got over. So we are going to continue from there in today's class. Just a minute. So in the last class we are talking about cloning vectors. Okay, in the last class we are talking about cloning vectors. In cloning vectors we are discussing about key features of the cloning vectors, in which we have taken the example of PBR322. Okay, plasmid PBR322. Okay, so in that the main features that a plasmid need to be have is, first we have discussed about cloning site. Yes, sorry, that is your origin of replication, that is ori site. Ori site, that is origin of replication. Then selectable marker, then your cloning site, okay, and after that actually we perform some test to check the success of transformation. Okay, so in the last class actually we have completed with the ori site and selectable marker, but still in today's class we are going to have a small glimpse of revision. Now ori site is actually origin of replication. I told you in the plasmid they have the capacity to replicate and increase their copy number. So there must be some particular site from where the process of replication takes place. Okay? Plasmid is also a double stranded structure, double helical structure, right? So actually this is the sequence. Okay, ori site is actually small sequence or site where the process of replication starts. First one. Second thing is that it is the region where we can also insert our desired gene. So during the replication, when the number of cells or number of the plasmid going to be multiply or increase, the desired gene also going to be replicate or multiply with that. Second thing we have discussed about selectable markers. See, selectable markers are actually a sequence of genes, or we can say small sequence, which have the capability to show resistance against some particular kind of antibiotics. Okay, selectable markers are the coding and coding for resistance for the particular kind of antibiotics. In the PBR322, we have seen that is the plasmid have the resistance against tetracycline. Yes, tetracycline and ampicillin. Tetracycline and ampicillin. Okay, so they show resistance against these two. They show resistance against these two antibiotics. So why it is necessary? Because see, we all know that whenever we need to form our DNA, recombinant DNA, okay, so they need to be transferred in the host. So we need pure quality. Yes, because if it is going to be mixed or impure form, then it may be the quality may not be good, or our desired gene transformation process may not be successful. So to check the purity, what we do the selectable marker. We use, or they have selectable markers, so that the particular resistance type of plasmid only going to be grow in that particular culture media which is containing this selectable marker or this particular kind of antibiotic. So we have already discussed about this these 2 points. Now the next one actually that we are dealing the last is your cloning site. Now what is cloning site? Cloning sites are that sequence of the plasmid where we are going to integrate our desired gene. Okay, we are going to integrate our desired gene because it is the cloning site where we are. It is the cloning site where we are going to use the restriction enzyme. Restriction enzyme. Restriction enzymes to cut the sequence on plasmid. Cut the sequence on plasmid and to integrate our desired gene. Now, what is the very much important thing with this point, cloning site, is that it will be very much good and beneficial if there is a single cloning site going to be present in the plasmid. Why? Because see, if the cloning site is going to be more than 2, I mean to say the recognition site for the restriction enzymes. We all know that restriction enzymes are specific with their recognition site. So, if there will be more than 2 or 1 recognition site, okay, many recognition site, what will be happen? That restriction enzyme is going to be cut in the multiple position, yes, and they may cause the generation of many fragments. So, what will be happen that when we are going to insert our desired gene, okay, and going to ligate, that there we are going to face problem. To solve out this, it will be very better if we choose a plasmid which is generally have single or very less number of recognition site. Okay, now see, I told you that restriction enzyme is going to be cut in a particular region. In the case of Urea 322, if you see the structure of the plasmid, I told you that they have the resistance against 2 types of antibiotics. Yes, they have the resistance against 2 types of antibiotics. One is resistance for tetracycline. Okay, one is resistance for tetracycline and another that is ampicillin. Got it? What we do, that we use the restriction enzyme, and that restriction enzyme is specific to cut this point, that is responsible for tetracycline, and this one is for ampicillin. Okay, that one for ampicillin. So what will be happen? Suppose that we use a single restriction enzyme or same type of restriction enzyme to cut to cut in plasmid, and also to withdraw or to cut our desired gene. I mean to say that we have to obtain a desired gene from another organism. Yes, so there also we use the same restriction enzyme, so that it is going to be cut at that particular point, and in case of plasmid also we are going to use the same, so that the flanking region or the end region which are going to be present or which are going to be formed should be common, so that the end which is going to be produce sticky end, that may going to help when we are going to transfer our desired gene inside the plasmid. Okay, the flanking region or the sticky end sticky end will make them help to join with the help of DNA ligase. So what will be happen? Once we use the restriction enzyme and we cut out this particular sequence, suppose that the restriction enzyme is going to cut here. Now what will be happen? After removal, okay, after removal of this sequence, okay, and then we integrate our desired gene which is going to be fixed here. Okay, our desired gene which is going to be fixed here, integrate here. Now B8 DNA ligase that is going to be joined here. Now what change you will see in this plasmid? Naturally this plasmid was having resistance against ampicillin and tetracycline antibiotic, but after treatment with the restriction enzyme, the sequence which is responsible for tetracycline resistance is no more. Yes, Instead of that, we are having our desired gene. Now, this is actually what our recombinant, or we have created our plasmid. We have created our desired plasmid which is need to be transferred in the host. Now what next? We transfer this to the host. Now we are going to grow the host, okay, in a culture media which is containing ampicillin. Reason behind that, because we know that the new plasmid that we have constructed have the resistance only against ampicillin. Okay, so what we do? We create a culture plate. Okay, this culture medium contains antibiotic, that is ampicillin. Now what will be happen? The plasmid which is there, they have the resistance against ampicillin, so they are going to be grow in this culture medium very properly. Okay, they are going to be grow in the culture medium which is containing the ampicillin, because they have the resistance against ampicillin. Now what we do? We take a small sample inoculum from this culture plate and again grow that in a culture medium which is containing tetracycline. Now, while we are doing so, to check the purity or to check the success of our transformation, because I told you that during the treatment with restriction enzymes, this sequence that is responsible for tetracycline is no more. So, in this, when we are going to culture, there will be no growth. There will be no growth of the host or microorganisms, because they don't have tetracycline resistance. They don't have tetracycline resistant antibiotic. Okay. But if it is going to be grow in tetracycline medium, it's confirmed that our transformation is not successful. Still, the wild type is present. Okay, so in this way we can check whether our transformation is successful or not. First point. Okay. Second thing, what we are doing, we are with the same thing. We have to do two plating. Yes. One for confirmation and one for the negative test. Yes. So it is time taking also, plus money wasting also. Lots of things are there. So we scientists have developed a new method. Okay, new method to check the transformation. Means transformation is successful or not. Okay, this means the plasmid which we have incorporated in that transformation is done or not. So what was that? You can say that blue-white screening. Okay, one method that is known as blue-white screening. Okay, that is blue-white screening, and one more, that is enzyme inactivation. Enzyme inactivation. Okay. Or you can say insertional inactivation. Insertional inactivation. Blue-white screening and insertional inactivation. There is not much difference. The concept in both the cases same. So we are going to see what is next. Okay. Now, till now we got our experiment done. In that we have integrated our desired gene into the plasmid, and we checked that for transformation. Now the next I told you, for the same, for the checking the purity or for checking the success of transformation, we are doing one more, that is blue-white screening or insertional inactivation. Actually what happened, if you are going to talk, suppose that example, you take the example of E. coli, okay, they have plasmid. Now they have the wild type, okay, the wild type have the capability to consume chromogenic substance and metabolize that, okay, get breakdown, they are going to break down the chromogenic substance. What actually they do in the blue-white screening or color screening, the culture medium, okay, which is containing the nutrient for the growing microbes or hosts, they have chromogenic substances, okay. So the wild type means not engineered one. Wild type have the capacity to break down the chromogenic material and release the color. They are going to emit blue color, okay. They are going to emit blue color. But the transform, okay, the transform will, they don't have that particular enzyme which is going to be break down that chromogenic substance. As a result, they are going to show white colony. They are going to show white colony. So the white colony is actually, they are the transformed one. In this way, we are going to identify our transform and non-transform one. So what is the thing? In this method, beta-galactosidase, okay. Okay, beta-galactosidase enzyme is responsible for lactose metabolism. Okay, and when they are going to break down lactose, they produce this is the case of non-transformer. But in the case of transform, means in transform, that is in which we have insert our desired gene, okay, in which we have the plasmid in which we have inserted our desired gene, they lost that sequence, that they, listen carefully, they lost that sequence which is responsible for synthesis of beta-galactosidase. That is the reason why we are saying that insertional inactivation. We are saying that insertional that is insertional inactivation. So what happened in this beta-galactosidase enzyme is not going to be produced, okay, which is not going to be produced. As a result, they are not going to be metabolized, they are not going to be break down the lactose. As a result, the chromogenic substances which is there, they are not going to be break down. As a result, no colorful colony, okay, or we can say white colony, okay, white colony. So these are the transformed, okay. In this way, in a single plating, easily we are going to identify, suppose the white colony you will get, okay, you will get white colony, and suppose you will get blue colony. Okay, so you will get blue colony. So what will be happen? These blue colony are actually white type, okay, that is not our desired plasmid, but white colony, okay, the white colony is actually that one which we have transformed, and again we take small inoculum from them and we culture them separately in different plates, and we will got all the pure transform one. Okay. So in this way, cloning vector help us to form the desired desired vectors with the particular characters. And yes, this is the case of microbes. Nowadays, in the case of plant and in the case of animals, also vectors are using. In the case of plant, it is Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens. They are specific plant pathogen. Okay, they are plant pathogen which is going to cause infection in the plant, causing plant cells to become cancerous. As a result, what happened? Continuous cell growth or cell division takes place in that particular region, causing tumor formation. Okay, causing tumor formation. Okay, that is known as TI plasmid. That is known as TI plasmid. That is tumor-inducing plasmid, and through which we can easily incorporate our desired gene in the plant. Okay. Agrobacterium tumefaciens. They are also called TI plasmid because they are going to cause tumor. Okay, in the plant, and through where we can easily incorporate our desired gene. So that is the case of plant. In the case of animal, okay, in the case of animal cells, it is actually retrovirus, which is used as plasmid, okay, and which in fact, and that also cause the same thing, okay, and through which we insert our desired genes. Okay, so TI plasmid with the help of Agrobacterium in plant, retrovirus used as plasmid in the case of animals, and all other features and characters we have discussed about the cloning factor. So by this topic, we are almost going to complete the chapter biotechnology, processes and principles. Okay, and again in the next class we will be there with the next video. Till then, best of luck to you all, and yes, thank you. Have a nice day.